Hello, this project is a potential Floridian environments and bioremediation and aiding against plastic pollution, isolating oil-eating microorganisms from three different environments. Oil and plastic are both some of the world's most used substances today, with over 100 million barrels of oil being used every day, and half of all material packaging being made up of plastic. However, they are both inherently harmful to living beings in the environment due to their toxic properties, and with thousands of oil spills occurring in the U.S. annually alone, with some reaching over 1,200 kilometers of shoreline, and with over 23 million tons of plastic being polluted in marine environments each year as well. Due to the severity of plastic and oil pollution, there are active efforts in cleanups through manual and chemical methods. However, they often lack funding, staffing, and can further contaminate the environment with chemical usage. It's been found that up to 80 general bacteria use the hydrocarbons from oil as a carbon source, feeding and growing from them, and degrading the oil as well. With bioremediation, or just another name for the use of bacteria in degrading oil, it was found to be cheaper when cleaning up 120 kilometers shoreline after the Exxon Valdez disaster in 1989, one of the worst oil spills in recent history when compared to manual methods. These bacteria can also produce bioplastics like PHAs and PHBs, which are biodegradable polymers being used currently in the medical field to treat injuries. So, microorganisms have been looked at as a cheaper, sustainable, and potentially more efficient alternative to current methods. And due to Florida's biodiversity in unique marine environments and the lack of studies of them for bioremediation, this project focuses on seeing what potential lies in local environments. In this project, the microorganisms were collected from three different environments: from the Everglades, from a mangrove environment, and from a beach environment, which was Naples. And the results and conclusions were based on the amount of colonies isolated per environment and the amount of gram-positive and gram-negative colonies. The main question is whether Floridian environments have microorganisms able to use diesel as a sole carbon source, and it was hypothesized that all environments would be able to have microorganisms isolated as they were able to use diesel as a sole carbon source. In the methodology, 500 milliliter water samples were collected from each environment at these geolocated spots. The three collection sites specifically were the Naples Public Beach, and for the Everglades, it was the Arthur Marshall and Loxahatchee National Refuge, and for the mangrove environment, it was the John Pennekamp State Park. Then, a bioremediation solution was created with the microorganism solution sample, which was again a water sample, diesel, and mineral salts media. And with the mineral salts media, it lacks a carbon source and lots of important salts. So the diesel added would be the only carbon source, and in order for any bacteria to grow, the bacteria would have to use the diesel as a carbon source. And then the solution was let to incubate, and then it was plated in order to isolate the bacteria grown and then organized into master plates. A gram-state assay was also done on one colony of each visually distinguishable group of microorganisms isolated in order to obtain some characteristics about the microorganisms collected as the strains and the types of microorganisms collected are not so far not able to be exactly identified. It was found that all environments had microorganisms isolated, with many colonies having different visible features and also amounts between the environments. It was found that over half of the colonies isolated were from the Everglades environment alone, at around 265, and with the mangrove and the Everglades environments having half, respectively, of the Everglades environments, and again having similar amounts to each other. And with the gram-negative and gram-positive data, it was found that the gram-negative colonies were found to be 8 to 3 to gram-positive, so again, overwhelming gram-negative colonies. And since all bacteria, and since all environments had colonies isolated, the hypothesis was proven, and all environments and microorganisms isolated will be worked with in the future research in the future, which will include testing the rate of degradation of the oil, the amount of bioplastics produced, and sequencing to identify exact bacterial strains. And this is done in order to determine the potential Floridian environments in aiding against plastic and oil pollution. Thank you.